The main disclosure is that I don't look at the neuro ITU, okay? So this will be quite simple to understand with just one or two take-home messages. I'd like to thank Claudio for bumping me to the bottom of the programme, but of course for inviting me to Vicenza. And as you probably noticed, he made me feel at home by importing the UK weather, so that's very kind of him. Now, when we lecture in the UK, we get graded, and so we get reports back to tell us how well we've done. And this is a report I got a couple of months ago from the College of Anaesthetists. So it looks fairly flattering until you look at it quite closely. And it says that my talk was not useful, I had a very poor presentation, and I looked bored while I was speaking. So you have my permission, if I look bored, to come up and poke me. So, best of friends, kidneys versus the brain, okay? So, are they best of friends or are they enemies during an insult? Now, we know that the kidney and the brain talk to each other. So we have neurological pathways, and as Ray Van Holder and Norbert Lemire discussed in his elegant editorial based on some mouse work, as they called it, Marconi revisited from kidney to brain, to organs communicating at long distance. And we know there's direct neurological involvement between the brain and the kidney. And we also know that this combination maintains our homeostatic milieu. And we all learned this at medical school about the effect of the pituitary and ADH, etc., etc. Okay? I'm not really going to talk about that. What I'm going to talk about is the effect of AKI on the brain. But before I do that, I'll just talk the other way around a little bit. Because if you develop acute kidney injury on top of neurological disease, you're in a bad, bad place. So if you have traumatic brain injury and you develop AKI, your hospital mortality goes up considerably. As it does if it complicates acute stroke, and as it does if it complicates subarachnoid hemorrhage. So getting renal failure or acute kidney injury on top of a neurological insult somewhat careless because you'll be in a very, very bad place. And this is, even though this is 2012, you can see renal failure was called renal failure. The interesting thing about this paper is they've actually used the GCS score in the right population. We all use the Glasgow Coma Scale. It's only validated in traumatic brain injury, but we kind of forget that bit. So there's much, much more to the story than just the brain being sick and then upsetting the kidneys. As you can see by this booklet here, kidney disease gave me brain damage. And so what we're going to briefly consider is how AKI affects the brain. Some of you may share my love of this particular movie that I think is Young Frankenstein with comic genius, but still. Here's the effects of acute kidney injury, as I see it, on the brain. There's the effect of the inflammatory cascade, which we've heard lots about today. The impairment of the blood-brain barrier, and that's key to what happens after. The derangement in neurotransmitters. Acid-base disturbance. Don't forget, it doesn't only affect the heart, it also affects the brain. The osmolyte disturbance, which has been alluded to, and importantly, altered drug metabolism. And all these concepts interact with our patients with acute kidney injury who then develop brain problems. And we know that acute kidney injury is a systemic disease. We've already heard that this afternoon, really. And so we have this varying degrees of organ crosstalk. One of those organs, intimately, is the brain. Now, I was just going to talk a little bit about the systemic inflammatory response in AKI. We've heard about this today, and this was actually a very interesting paper. And it sums up the inflammatory response in AKI as three main signals. You get a surge in uric acid, or the DAMPs, a secondary response, and then a toll-like receptor in activation. So here we have this surge in uric acid, which is our initial burst, where the systemic inflammatory cascade is triggered. This in itself may, of course, in the future lead us to some therapeutic window, because if the uric acid, for example, or an equivalent, triggers a cell cycle arrest response in the kidney, we may be able to almost inoculate the kidney against further damage. That's another talk that I haven't written yet, so we'll save that for next year. So you get this surge in uric acid, then exocytosis, and this dramatic inflammatory process, including
clotting, and this production of these Weber-Pollard bodies that weren't described when I was at medical school, but now are a hot topic with von Willebrand factor monomers and intravascular coagulation. And then that leads to the later stage of the release of HMGB1, etc. So what happens to the blood-brain barrier? Well, what do? Your systemic inflammatory response damages the endothelial permeability, and particularly in the floor of the third ventricle, allows passage of the inflammatory mediators into the brain. Now, that particular area, OTLV1, is like the tongue of the brain that tastes everything that's going on. So when we develop hyperpyrexia, for example, that's the bit that senses it and triggers whatever response. So you get exactly the same in a systemic inflammatory response. OTLV1 senses that, and then you get systemic inflammatory response, and the blood-brain barrier increases in permeability. And that has been shown in animal models. You'll be pleased to know that most of the stuff I show with brain biopsies all comes from animal work. There's very little human data. So the response in the kidney and the brain seems to vary in terms of the inflammatory picture. But what we do see in AKI, this is in mice that have had their renal arteries occluded, we get an increase in expression of various proteins associated with brain damage and fibrosis. And this is glial fibrillary acidic protein, GFAP, which is expressed in astrocytes when the animals have been exposed to a period of acute kidney injury. We also see extravasation of dye. So this is Evans blue, so it's a marker of cellular integrity and membrane integrity, and with AKI you get movement of albumin across the membranes that's quite florid. So you get this increase in the blood-brain barrier permeability. It's also seen in this study, rats in this case, where they're given inoculums of E. coli, and you can see there's a dose-response curve increasing your blood-brain permeability. So acute renal failure, stroke sepsis, stroke inflammatory process gives you an increased blood-brain permeability, and then, as shown by this slide here, and then subsequent complications. So AKI, inflammatory response, disruption of the blood-brain barrier, endothelial injury, and stimulation of the inflammatory and coagulation cascades. Now, as I alluded to, you also get changes in neurotransmitters, and the precursors of neurotransmitters, again, are closely regulated by the channels deeply set within the blood-brain barrier. And amino acid transporters are particularly important for this. And CAT1, SLC7 are two types of amino acid transporter. They regulate arginine, which is a precursor for taurine, again, a major component of neurotransmitters within the brain. And so you may get accumulation of some precursors and yet depletion of others. And that has been shown again in animal models where uremia suppresses central dopaminergic metabolism. And this was work done in rats, again when I had renal failure through an inclusion model. And they looked at rat activity in uremia compared to the sham. And the uremic rats unfortunately didn't do very well and were very motionless and feeling somewhat lethargic, which no doubt most of us are after the day we've had. And here we see the dopamine levels within the brain, and those exposed to acute tubular necrosis in this model had reduced dopamine levels. So what does that mean for our patient? Where does our patient sit here? Well, this is a cartoon describing delirium in our ITU patients. And you can kind of pick out the bits that AKI would tick, really. Observable and occult metabolic abnormalities, we know we see that in acute kidney injury. Lack of homeostasis, that's particularly important in acute kidney injury. Medical comorbidities, most patients with AKI have a degree of comorbidity. Uremic encephalopathy, major interference in terms of delirium, and of course on top of that, you can get coma, but in fact occasionally with uremic encephalopathy you will get hyperexcitation. Again, AKI causes acid-base disturbance. What does that mean to the brain? Well, it can affect glutamate dehydrogenase activity, which is involved in neurotransmitter balance, and there's direct acidifying effects on the CSF itself. The osmolytes, okay, your osmotic active agents within the brain, retention of nitrogenous waste leads to increase, particularly in urea concentrations within your astrocytes and neurons. Professor Gibney spoke about this earlier, and an increased intracellular hypertonicity leading to cell swelling. One of the things we see quite commonly, of course, is the effect of dysnatremia and acute kidney injury with its effect on the brain. And this is a very good study from critical care, Michael Darman and colleagues in France, that looked at minimal changes in sodium levels and showed quite an extensive problem in terms of outlook. I'll quickly whiz through some of the sodium abnormalities we can sometimes see in AKI affecting the brain. Cranial diabetes insipidus, we see post-traumatic brain injury and is apparent as a dysnatremia. Patients are initially very polyuric with a highly dilute urine that can sometimes become transiently oliguric later on in their stay. But the most important problem with these patients is if you don't realize it, the patient may develop profound life-threatening hyponatremia because in the ITU they quite often can't drink because we don't let them. Hyponatremia is another issue which can affect the kidney and the brain, occurs following acute cerebral damage in many cases, and your mortality significantly falls, as does your serum sodium. Then there's nephrogenic sodium wasting, which you probably know as cerebral salt wasting. It's been renamed fairly recently, as you know. And sometimes clinically, it's difficult to distinguish between the two. In SIADH, patients are typically euvolemic, whereas in cerebral salt wasting, their volume deplete. Biochemical investigation may be unhelpful in that case. It's important, of course, to make the correct diagnosis because patients with nephrogenic salt wasting need a lot of salt in a fairly controlled way and fludrocortisone, whereas the treatment for SIADH is almost the absolute reverse. I'm sure you get these slides coming with the CD, so this gives you a treatment diagram which is, you can apply at the bedside. So finally, drug metabolism and the effects of AKI on that, downregulation of organic anion transporters. These are involved in transport of exogenous chemical compounds as well as uremic toxins. And there's been some fairly good work on the effects on OATs in the brain following acute kidney injury, again, all in animals. The other thing is, of course, that sedatives and opioids have significant renal clearance and therefore there's an increased half-life in drug accumulation. There are specific things you can see, like penicillin encephalopathy, where you get inhibition of OAT3 and you can get a build-up of penicillins in that case. Quick word about uremic encephalopathy. As we've heard, it's an organic acid accumulation in the brain. We heard an elegant talk about the metabiome, and there were hundreds of candidate molecules there. This results in changes in both neuronal intracellular osmolality as well as transmitter levels. You can get these classic EEG findings. Here they are. I always have to write down what they are at the bottom because I can't tell you what they are. So it's loss of alpha and beta waves. And when we get EEGs done on our unit, I make sure the technician's there to tell me what they mean. So finally, a quick apology for my bias. We're supposed to link this into renal replacement therapy. And of course, we all know in our patients with uremic encephalopathy, then intermediate therapies are probably not the best. And this was a very elegant study by Claudio in the last millennium. And hopefully you've all taken home the messages from that in that patients with marked uremia do less well with intermittent therapies. And this is data from Andrew Davenport, who in fact should be giving this talk, but he's in London at present. This is the effect on intermittent hemodialysis on the ICP. And as you can see in patients with uremic encephalopathy, there's quite often a marked rise. The mechanism for that is, of course, you get increased urea, then you get rapid removal of plasma water, delayed clearance from the CSF and the neurons, sets up a urea gradient between the cells and the plasma, water enters the brain, you get worsening cerebral edema, and you end up with a picture like that. And if you're unfortunate, a dead patient. So in conclusion, sorry that was a bit of a whistle-stop tour, but AKI increases cerebral inflammation, impairing the blood-brain barrier, resulting in functional deficits, disturbed neurotransmitters, acid-base disturbance, osmolarite disturbance, or to drug metabolism. Take-home message, if you use intermittent hemodialysis for uremic encephalopathy, don't say you weren't warned. And with that, I thank you for your attention.